All right, so this video is for section 6 of chapter 4, and we're going to talk about fermentation. So right away, I need to know a definition for fermentation. Essentially, it's an anaerobic process that recycles NADH back into NAD+, so that glycolysis can continue, right? One of the things that we haven't talked about yet is that when we have glycolysis, so this first part of this picture, and this needs to be in your notes, is actually just glycolysis. We've seen this before, right? You have a 6-carbon molecule, this glucose, that's going to be broken down to make 2 high-energy 3-carbon molecules, 2 pyruvates, right? And in the process, you get to capture some energy. You get 2 net ATP out of that, which is great because I can use that energy in my cells, right? So I make 2 ATP, but I also am going to make 2 NADH molecules in the process. And one of the things that we haven't talked about before is that, right, all 3 of these things, the ADP, the glucose, and NAD+, are reactants for this process. Without ADP, you can't make ATP, right? That should be pretty obvious, right? So it's possible that I could run out of ADP in a cell, but that would actually be a good thing, not a bad thing, because the only way that I can replenish that ADP is by actually using energy, using that ATP for some purpose, and that should happen pretty frequently, right? If I did run out of ADP, that just means I have too much ATP floating around in that cell, right? More than I could possibly be using. So that's never really a bad thing. Just like it's not really a bad thing if I have too much glucose, right? I could run out of glucose, and that would be a really bad thing. So there are some people that have a hard time moving those glucose molecules into their cells. I forgot a 6 here. And they need insulin, right? They need to take insulin so that they can get that glucose into their cells like they need. But otherwise, assuming you have no problem getting glucose into your cells, assuming you have eaten something in the last day, you're probably not real low on glucose in your cells. So ADP, if I'm low on that, that can only be a good thing, right? Glucose, we're going to assume that you have plenty of that in your cells or you have other problems. But this NAD+, it is actually possible to run out of that NAD+, right? When we talked about our electron transport, I'm supposed to use this energy in this NADH molecule. I'm supposed to drop off electrons in my electron transport, but electron transport doesn't work without oxygen available. When I don't have oxygen available, I'm going to make a bunch of these NADH molecules, but I'm never really going to replace those NAD+ molecules in the cell. And those are going to start running low. And what may not be obvious is that all 3 of these reactions, the glucose turning into pyruvates, the ADP turning into ATP, the NAD+ turning into NADH, all 3 of those things have to happen for this process to work. I can't make ATP if I'm not going to make NADH, right? If I don't have something to catch electrons in this reaction, this glucose turning into pyruvates, then I can't do any of it. I can't change the glucose into pyruvates. I can't make the ATP. I'm just stuck. So if I run low on this NAD+, I have to find a way to replace it. If I'm not going to use the electrons in NADH in electron transport, I have to have another way to recycle those molecules to replace that NAD+, or else I'm in trouble. So this fermentation reaction, this arrow that I'm drawing here, is the first part of this that is actually fermentation. And it's a simple process. I'm going to change those 2 pyruvates into lactic acid molecules. Right? Lactic acid has 3 carbons just like the pyruvates, so I didn't really change much. I just rearranged some things. But I'm going to make 2 lactic acid molecules, And in the process, I'm going to get these two NADH molecules to turn back into NAD+, right? Changing pyruvates into lactic acid is actually a catabolic reaction. And it's a wasteful catabolic reaction because I'm losing those electrons in NADH. I'm wasting those electrons doing just this side reaction because it's really, really necessary for me to replace this NAD+. I don't actually want to do this reaction in my cells because the lactic acid itself is toxic. It causes problems in the cells, right? You know about that because you've been sore before. Lactic acid in your muscles, a buildup of lactic acid can cause issues, right? It hurts for a reason. It's actually a bad thing in your cells. You can get rid of it pretty easily. Not to say that it's a bad thing to work out and get sore, because sometimes that means that you actually accomplish something, right? But it's not actually a good thing in your cells. You can't make too much of it or you can have issues. But some way, somehow, I have to replace that NAD+ that's key to this glycolysis reaction. So it's actually pretty simple. The picture is not hard to draw. It's just kind of hard to understand, right? So I have to worry about these three reactants. Again, if I ran low on ADP, that would just mean I have lots of ATP. So that's not actually a problem. If I ran low on glucose, that could be a problem, but that's a separate problem, right? Maybe I am a diabetic and I have a hard time getting glucose into my cells. That could be an issue. But assuming I have glucose in my cells, assuming I have eaten something recently, right? The only thing that's going to run low that's really a problem is this NAD+. So again, without the NAD+, I can't make the ATP over here. So in order to make ATP when there's no oxygen available, I have to somehow replace that molecule. And this lactic acid fermentation, I change the pyruvates into lactic acid molecules with the purpose of making NAD+, so I can continue glycolysis. Otherwise, the lactic acid is toxic. I have to get it out of my cells. I don't really want it in my cells. The only reason I did this is because I needed so badly to replace NAD+. So this is what happens in your cells, right? When your muscle cells are without oxygen, you have to do this fermentation. One thing you might add to your picture, make sure that you can see that like this side is actually just glycolysis. So it's not different than what we talked about before. This side is the actual fermentation. So keep those things separate. There's just one little reaction. I change the pyruvates into lactic acid, but I do it so I can replace the NAD+. Alcoholic fermentation is pretty similar. I still have glycolysis. I'm still going to split the glucose in half to make two pyruvates. Sorry, this is a U. Might not look like one, but those pyruvates are made just like normal. I am doing this with the purpose of making some ATP. I'm assuming that the cell is in anaerobic conditions, so I have to continue doing glycolysis. All the ATP I'm going to get comes from glycolysis. I'm not going to do any more extra steps. And the main reason I'm not going to do those extra steps is because then I would just run out of this NAD+ even faster. So this is the same picture as it was before. NAD+, right, can be turned into NADH. All three reactions have to happen. If you want to make ATP, you have to make NADH. But same as before, if you run out of NAD+, you're in trouble, right? You can't continue to make any more ATP, so you have to be able to replace that molecule. So any kind of fermentation exists just to replace NAD+. I'm going to waste these pyruvates. I'm going to turn them into some different products, right? Let me make that arrow a little cleaner, right? I'm going to, in this reaction, I'm going to make some ethanol molecules, and I'm going to make 2 ethanol molecules. Now, this molecule only has 2 carbons in it, whereas the pyruvate has 3 carbons. So if you guys remember from before, that means I have to have kicked out something else as well in the meantime. So I'm going to have a CO2 byproduct. I'm going to make 2 of those. For every 2 pyruvates, I kick out 2 carbon dioxides and make 2 ethanol molecules, right? The ethanol itself is the alcohol that we're talking about, right? And that is toxic. You guys know that alcohol is toxic, right? This is the alcohol that does have recreational purposes. If you were 21 and older, you could drink that, but no one should drink excess amounts of that because it's actually bad for your cells. It'll kill your cells. It's toxic, right? This does not happen in your cells. Let's be very clear. This happens in other microorganisms like yeast, right? They will do alcoholic fermentation, whereas you do the lactic acid fermentation. Completely different, right? You make lactic acid when you're working out and your muscles are starved of oxygen, but other organisms have other pathways that they'll do, right? The whole purpose of this reaction, changing those pyruvates into ethanol, was to recreate this NAD+. Remember, we're running low. Otherwise, both byproducts over here, the carbon dioxide and ethanol, are toxic, right? Again, carbon dioxide is a normal thing in cells during respiration in general, so that's just gotten rid of like normal. But those ethanol byproducts, those become a problem pretty quickly, right? So a cell can't do this for very long and survive. But when that cell is starved for oxygen, when that yeast organism is starved for oxygen, it'll do what it has to do to stay alive. Now, the reason we go through this, again, is not because it happens in your cells, but because these byproducts can be useful for us, right? And I'm not talking about this as being useful in a recreational purpose. When we're talking about ethanol on a test, the reason we care that ethanol can be made is because we can use it as a fuel additive, right? You've seen that on the pump. You know that there are fuels that have ethanol in them that burn more efficiently, more cleanly, right? And it's cheaper because you put that ethanol in there. So that's a good thing, right? You also are wearing synthetic clothing, probably. And that can only be made using these 2 carbon molecules as a carbon source, right? You're going to make these polymers, these long strands, these big molecules that are actually, they're going to come from ethanol ultimately. So if you're wearing some kind of synthetic clothing, if it's not 100% cotton, chances are ethanol was an important thing to make that piece of clothing. So there's lots of other alternate uses for this ethanol aside from recreational purposes, right? Since you guys are not 21, you don't get to answer that way on a test. You have to talk about it being a fuel additive. You got to talk about how it can be used as a carbon source to make fibers for clothing. Otherwise, carbon dioxide is a really important byproduct for this because you should know that, again, this is happening in like a yeast cell. Excuse me, forgot the S. And yeast can be used to make dough rise as a leavening agent. And it's this carbon dioxide gas that creates the bubbles that make that bread dough rise. All right, so those are our 2 pictures. That's all you really need. Make sure you know the context, why we would talk about this alcoholic fermentation. Otherwise, lactic acid fermentation happens in your cells.